Okay, right, so we have the last lecture in the series, from me anyway, and this is going to be on bacterial pathogens. So we'll start out with some general characteristics of bacterial infections, and we're only going to look at two specific examples, 1 gram-positive infection and 1 gram-negative. And then I'll end up with a little bit about the genetics of virulence. So to start out, it's always good to, well, maybe not always good, but in this case it's useful to go right back to the beginning, to the 19th century, when Pasteur and Koch and a couple of other microbiologists were trying to show that bacteria were responsible for human diseases. And the way to prove that any microorganism is causing a pathology was set out by Robert Koch with his famous postulates. So he said that you can really prove that your microorganism is causing disease if you find it in all patients with the disease and it's absent from healthy people. And within the patients who are infected, you can find the microorganism in the organ which is affected by the pathology. Now you might think that that's pretty much all you need to do. If your microbe is present in the organ of all people who are sick, it's not present in people who are healthy, that's it. Yeah, case closed, thank you. But no, this is enough to establish a correlation. You've correlated the presence of the bacterium with the pathology, but that doesn't demonstrate causality. And this is really the heart of the debate between Louis Pasteur and the members of the Academy of Medicine. So the physicians in the Academy of Medicine said, look, Pasteur, you idiot. Of course you're finding bacteria in these sick people. They're sick, right? That's why they have bacteria. So you might think this is a kind of obtuse way to reason, but it's not necessarily false. For example, people with cystic fibrosis, you can find very frequently they have infections with Pseudomonas aeruginosa. It's one of the leading causes, one of the leading life-threatening infections in people with cystic fibrosis. And you don't find very much Pseudomonas aeruginosa in the lungs of healthy people. So if you were to just establish the correlation as, it's Pseudomonas aeruginosa infection which is causing cystic fibrosis. That's wrong. You've got the causality the wrong way round. Okay? So in order to establish that the bacteria are causing the pathology, you've got to go a bit further. And you have to perform a controlled experiment. Okay? And this is where the next set of Koch's postulates come in. So you have to isolate the bacterium, propagate it in pure culture, and then if you inoculate that bacterium into a healthy subject, most often an animal, right, then it should induce the same disease symptoms. And this way you can prove causality. And then really if you want to get, it's the cherry on the top here is you should be able to re-isolate the same organism from the experimentally infected subject. And if you can perform all of these experiments, you've really proven that your infectious agent is causing the disease. And this is true for bacterial infections, fungal infections, and viral infections. However, so this is the perfect case. If you can prove 1, 2, 3, 4, you've done it. But in a number of different cases, it's not actually possible to go through all of these steps. So if you have healthy carriers, You might not have a perfect correlation between the presence of the bacterium and the presence of pathology, because some people who carry the bacterium won't be sick. So this is something that was not known in Koch's time. Some bacterial infections can cause pathology in an organ where the bacteria are not present, because they can release exotoxins that are particularly damaging to an organ where there are some kind of organ in the body. So urohemolytic E. coli infections, they are localized in the gut, but the bacteria will release a large amounts of toxin that will damage the kidney. So here it's number 2 won't be respected, because if you isolate the tissue, kidney tissue from these people, you won't find the bacteria. Number 3, this means that you have to be able to propagate the agent in culture. Not necessarily feasible. So Mycobacterium leprae cannot grow in a culture system. So you can't get it in pure culture. This is also true for some viruses. Human papillomavirus is very, very difficult to cultivate. Also true for hepatitis C and noroviruses for a long time. So it's often very difficult to establish all of these 4 steps. So this is in the best possible case. So once you've done that, you can prove that your microorganism is causing disease. And once that's been established, then you can try and think about categorizing these different types of bacterial pathogens. So some of them are strict pathogens. For example, Mycobacterium tuberculosis, and others are opportunistic, like Pseudomonas aeruginosa. So strict pathogens are always associated with disease. They don't grow outside of the human body, and they are not part of the normal microbiome. Whereas opportunistic pathogens are either commensal bacteria, like Escherichia coli, Staphylococcus aureus. We can call it an opportunistic pathogen, because you often find these species in the gut or on the skin of people who are perfectly healthy. And rather less frequently, you have some saprophytic bacteria, so chemoheterotrophs whose normal mode of life is just degrading organic material. Some of these can colonize people who are particularly sensitive to those infections. So Pseudomonas aeruginosa will really cause pathology in people who are immunosuppressed or have an underlying lung pathology, like cystic fibrosis. So strict opportunistic pathogens. And what defines a pathogen compared to a commensal bacterium is virulence, the ability for the bacterium to provoke a pathology. So there's 2 main components to virulence. One of them is to help the bacterium establish an infection in a new host. And secondly, got to be able to actually, once it's there, harm the host. So this can be measured experimentally by the ID50 or the LD50. So ID50 is the 50% infectious dose, LD50 is the 50% lethal dose. So experimentally, what you would do was you would get a whole bunch of mice, for example, and inoculate mice with different doses of the bacterium. And then at some later time point, you would sample them again, see if the bacterium has established an infection. This will give you the ID50. And then at another time point, see how many of the mice in each group will die. Yeah. 50% infectious dose. So that is the quantity of bacterium that will be required to infect 50% of the mice. So if you've got a group of 10, you would have 5 out of 10 infected. LD50, you inoculate different doses of bacteria. It's the quantity of bacteria necessary to kill 50% of the inoculated animals. Okay, so 5 out of 10 will be killed at the LD50. So the lower the LD50 is, the more pathogenic the bacterium is, because you need fewer bacteria to kill the infected organism, right? And different strains of the same bacterial species can be more or less virulent. So Escherichia coli, it's normally a commensal. The LD50 will be very, very high. Some strains are pathogenic, their LD50 will be lower. Same for Staphylococcus aureus, same for Streptococcus species. Yeah, sure, yes. So commensal bacteria, all right? These will have a very, very low ID50 because they can easily colonize a human being, but their LD50 will be very, very high because they are harmless. So they've basically got like capacity 1, okay? So they can infect and invade new hosts, but they won't hurt us. So for pathogen, yes. Question? You're measuring 2 different outcomes. No. Oh, no. This is ID50 or LD50. Excuse me. Yeah, this causes confusion here. So this is not meant to be mathematical. It can never be, the LD50 can never be lower than the ID50. Because in order to kill the host, it's first got to infect the host. But you can have highly pathogenic bacteria or viruses. These will be close, right? These will be similar numbers. The less pathogenic the infection is, the bigger the difference will be between these 2 numbers. Yeah, this is an epidemiological problem for all highly pathogenic infections, and it's kind of behind the tendency for highly pathogenic infections to burn themselves out. Okay. Yeah. Okay. So the question is, if your infection is too pathogenic, how can it persist in the population? And that's true. Okay. So you've got your population, your bacterium comes in, it's going to infect and kill people. And often you have 2 outcomes. People are going to be killed, or they recover and they become immune. But so you'll have a whole bunch of people getting killed, and some people will survive and become immune. But in either of these 2 cases, there are no more susceptible hosts in the population. So at the end, your bacterial infection cannot propagate anymore. So one way or another, you need to have new susceptible hosts arriving somehow. So one of the ways this occurs is for newborn young children. Okay, you've got a birth rate in the population, so you're always having new susceptible hosts arriving because they're not immune yet. So this is the epidemiology of childhood diseases. Another thing that can happen is, maybe you don't just have a single host. So if you have a wild species, yeah, exactly. So this is what's going to be happening with plague, right? So if you have another potential host, then the pathogen can disappear from this population because it's either infected everybody or everyone else is immune. right? So there's no more transmission within this population. But it can be transmitting within another reservoir where the pathology, where the pathogenicity will be less. So it won't be killing so many people, it can be transmitted. And then generations later, this population has become susceptible again because all the people who have been immune, they're all dead. And you have a new population where everybody is susceptible. And then you can get transmission from the reservoir again and you get another epidemic. Well, I would say, okay, for the pandemics, yeah, when you have transmission of avian influenza to humans, yeah, this is where the pandemic strains come from. But as well as that, you have the seasonal strains that are always mutating and so can continue to circulate in the same population.
So ID50 and OD50 are a function of the host, right?
Yes, yes. So for the same bacterium, we'll have a different ID50 or LD50 depending on the host. So your pathogen can be more or less host specific. Okay, whether a pathogen can carry on circulating in a population indefinitely depends on the host population size. Okay, because of this idea that every generation you have children being born, but your population needs to be big enough for this to maintain transmission chains indefinitely. So if you've got a population the size of the Faroe Islands, then you have an epidemic will arrive, it will infect everyone, then it will burn out. But if you have a population of several 100,000, where there are links between all the people in that group, then the same infectious agent can carry on transmitting indefinitely. So, big cities, that's definitely enough to maintain transmission even of a very highly pathogenic agent, if it's infectious enough. Right, we're stepping over the bounds of the... yes, another question. Yeah, sure. The ID50 and LD50 are going to be dependent on transmission mode. So for the same bacterium, depending on the route of infection, this can be very different. So anthrax bacillus can cause infection transcutaneously and through inhalation of the spores, and the LD50 is very, very different in the two cases. I believe inhalation is more severe. Yeah. Yeah, so it depends on the route of infection. Okay, so I'm very sorry, there's no real infectious diseases epidemiology contained in this course. We're trying to, so let's get back to microbiology here. Okay, so if you break down these two aspects of pathogenicity, being transmissible and being able to harm the host, you've got a number of different steps or a number of different dimensions to this. Okay, so in order to establish an infection, you've got to have the bacterium's got to be transmitted, it's got to be able to colonize a host, and it's got to be able to grow in the host. So 1, 2, and 3 here. These are to do with the ID50 and being able to transmit to a new host. 4 and 5, these are going to be related to pathogenicity. Okay? Whether the bacterium can escape host defenses and can actually do something that will directly damage the host. 3, maybe 3, 4, and 5, there's a bit of an overlap here. Okay, so this, if you've got some bacteria that are growing wildly inside your body without any control, at the end it's going to be damaging. So there's a kind of overlap with pathogenic potential and infectious potential with number 3. Okay? So, but 1, 2, and 3, commensal bacteria need to do these things as well, right? Okay, so let's have a look at the different mechanisms. So just for basics again, just as with viral infections, all pathogenic bacteria need an entry route into the host. transmission can be direct or indirect. So direct transmission, one infected person has a direct contact with the next person who's going to be infected. Transmission can be indirect. So one infected person will contaminate something, and then that contaminated whatever it is will infect the next person. So anthrax bacillus is spore forming, dead animal will release spores into the ground, and that field will remain contaminated for years or decades. So indirect contamination through spores. Listeria monocytogenes. It's a foodborne infection. Somebody who's infected will have contact with, they like to grow on meat products. So pate, cold meat, this kind of stuff is kept in the fridge. It's psychrophilic, so it can grow at 4 degrees. So the next person who will eat some of this pate can become infected, even though there's no direct contact.
For example, would it be considered direct or indirected? Like, for example, someone who's infected another, let's say, an animal that doesn't present any symptoms, and there's that contact with the animal. Is that a direct or is it?
Well, okay. So if you've got a kind of living animal as the intermediary between you, this is like in parasitology, you have this as the definitive host and the intermediate host. So, yeah, it's like vector-borne diseases as well, the similar kind of thing you would say that in that case you would consider the other organism that transmits the infection is the vector in that case. And so you have transmission chains which go from the definitive host to the vector and then back to another definitive host. But this is slightly different because we're talking about kind of inanimate parts of the environment that can maintain the bacterium in an infectious state. Okay. So cholera is going to be excreted from an infected person, gets into the water, and then someone can drink the infected water. So routes of entry. Okay. Oral, so all of these bacteria that cause gastroenteritis, foodborne, waterborne infectious agents. Respiratory mucosa, so diphtheria, tuberculosis, people's lungs are infected, they will cough, and this is the mechanism of transmission from one person to another. Cutaneous infections. Just like in viruses, it's relatively rare for bacteria to infect the skin. We have time with this. So, typhus, transmitted by lice, is an example where you have bacteria deposited on the skin by the vector, and then when you scratch the bites, then this will help the bacterium to penetrate the epidermal layer and get into the skin cells where it can start infection. But this is quite a rare example. Okay, so skin is pretty rare. Transcutaneous, that's very frequent. So you'll get a cut or a bite and it becomes infected. So Clostridium, this is why you should keep up with your tetanus vaccination because you can easily pick up an infection from a cut or a scratch. Streptococcus pyogenes can ease, it's very, very widely distributed. You can be infected. You should disinfect cuts and scratches to prevent these bacteria getting a foothold. And just like with viruses, there are quite a lot of sexually transmitted bacterial infections. So Treponema pallidum causes syphilis. Neisseria gonorrhea, well, as its name suggests, causes gonorrhea. Okay, before we move on, this one, true or false? Everybody got an idea? Who says true? One person. Who says false? 4 or 5 people. Other people don't really know. It's false. Okay, yeah, exactly. So you have these kind of... It's true that it can only grow, yes. I would say healthy carriers can be affected by opportunistic or strict pathogens. So you have people who are healthy carriers of Salmonella, which is considered to be a strict pathogen because it is not a normal member of our gut microbiome. So in humans, Salmonella is a strict pathogen. But you can have healthy carriers. So getting back to the question that was up here 1 second ago. Yeah, okay, so it's true that strict pathogens will only grow actively in the host, right? But some of them can persist in a sporulated form. Okay, so that's the kind of reasoning behind this question. Okay, so those are different steps of the infectious process. Now, try and think a little bit about molecular mechanisms now. So, colonization, kind of like for viruses, the bacterium has got to adhere specifically to host cells or tissues. And this is going to be a function of bacterial adhesins, and there are 2 types. So, Fimbrial adhesins and kind of non-definition, which is non-Fimbrial adhesins, everything else. So Fimbriae. As you remember, filamentous structures on the outside of the bacterial cell can be present in gram-positives and gram-negatives, and one of their functions is adhesion, either to inert surfaces, but in the case of pathogenic bacteria, they can be involved in adhesion to host cells. Now, this can be very important for bacterial tropism, which site of the body is infected. So, for example, uropathogenic E. coli, which can infect your urinary tract, they express P. Fimbriae, whereas enteropathogenic E. coli express a different type of Fimbrial adhesin, which allows a bacterium to adhere to enterocytes. So the specificity of the adhesin is determining which site in the body gets infected. Okay, Neisseria gonorrhea, they also express a Fimbrial adhesin that allows them to bind to the urethra and the genital mucosa. So other types of Neisseria which do not express it as this adhesin will not be able to establish an infection at these sites. So it's an important virulence factor, the adhesin. Non-Fimbrial adhesins, okay, any kind of structure on the outside of the cell that is not filamentous. So Yersinia enterocolytica, the major adhesin is called YAD-A. So this is the structure here on the outside of Yersinia enterocolytica. Yersinia pestis uses another molecule as its non-Fimbrial adhesin, AIL. So, yes. Yes, yeah, they must have some kind of adhesin, otherwise they won't be able to bind specifically to cells. And some gram-positive adhesins are Fimbrial. They have Fimbriae, but they are similar to the curly type Fimbria that exist in gram-negatives. So, so Fimbrial, non-Fimbrial also exists for gram-positive infections. Okay. So the bacterium has arrived at the site of infection. It's got to grow somehow. Uh, Generally, a living organism is full of all sorts of goodies for bacteria, loads of organic carbon, loads of organic nitrogen. Everything is there. However, there's almost no free iron in extracellular fluids because it's all, or almost all, bound up in iron transport proteins. So any bacterial pathogen has got to have a mechanism to accumulate or gather or iron, and there are different mechanisms that are involved. Some bacteria express very high affinity siderophores that are able to scavenge the very small amounts of free iron that exist. So that's used by Mycobacterium tuberculosis and Yersinia species. Other bacteria, they think, okay, why, well, there's a whole bunch of iron floating around in the body, but it's just bound up with these proteins. So if I've got a receptor for some of these proteins, I'll just bind the iron transporting protein, internalize it, eat up the protein, and extract the iron. So pathogenic Neisseria use this mechanism. And of course, there's a ton of iron in our bodies in erythrocytes. So hemoglobin is a source of iron for many bacterial pathogens when it's associated with hemolysis. So these other pathogens can use hemoglobin as a source of iron. Now, of course, our bodies aren't sitting around waiting to be colonized by bacteria. Our immune system, we have a whole bunch of antibacterial mechanisms in the immune system. Probably the most important one is just phagocytosis and destruction of bacterial cells by neutrophils and macrophages. So a very important element of bacterial pathogenesis is to avoid being killed by phagocytes, the cells of our immune system that are going to arrive at the site of inflammation and internalize and kill bacteria. So some pathogens try to avoid being phagocytosed entirely, either by having a capsule antigen. Very important. So this is going to cover up the outside of the bacterial cell and hide the structures that are recognized by receptors of the innate immune system. So it's going to hide peptidoglycan. It's going to hide LPS, which are conserved structures. At least the core oligosaccharide in LPS is highly conserved. And so if you hide these conserved structures behind a variable capsule antigen, this is going to make phagocytosis a lot less efficient. So all the pathogenic strains of Streptococcus pneumoniae, Neisseria, meningitis, Haemophilus influenzae have a capsule. Other bacteria will find a way to survive inside the phagocytes. So they don't avoid being internalized, but once they're internalized, they find a way to survive. So either they'll escape from the phagolysosome. So this is Listeria will do this, Rickettsia will do that, and they live inside the cytosol. Yeah. Rickettsia, I don't know actually, but Listeria monocytogenes expresses a protein called Listeria lysine, so it's going to lyse the membrane of the phagolysosome, and this will allow the contents to escape into the cytosol. And other bacteria will manage to survive inside the phagolysosome. Now, how can this occur? Normally, what is going to happen after phagocytosis, you have a maturation from early endosome, late endosome, and then a lysosome. And this maturation is dependent on acidification, the endolysosome. So the bacteria that manage to survive inside the phagolyzosome will prevent maturation. So somehow they will subvert the host phagocytic cell and prevent the acidification of the phagolyzosome. So this means that proteases, lipases, lysozyme don't get secreted into the phagolyzosome and they will not become active. So the phagolyzosome can become an environment which allows these cells these bacteria to survive. And finally, damaging the host. Okay, so in order to provoke pathology, pathogens have got to actively harm the host. Okay, so two types of process here. One is producing hydrolytic enzymes, and this is also to do with invasion. So many bacterial pathogens produce enzymes that are going to attack the extracellular matrix. So collagenase, hyaluronidase, elastase. So this is part of the invasive process because a lot of the time bacteria are going to be infecting epithelia. Okay, so you've got your bacteria that will start to colonize the epithelium and they'll kill some of these cells here. So they get access to the basal membrane. Now, if they want to get any further in the body, they've got to degrade this extracellular matrix. So that's what the collagenase, elastase enzymes are there for. So this will allow these bacteria to degrade the basement membrane, and then that's it. They can disseminate from the initial site of infection to somewhere else in the body. Now, other bacteria have a bunch of other hydrolytic enzymes that can be involved in pathology. So Staphylococcus aureus is very, very particular because it induces clot formation, blood clotting, and this is very important for the production of abscesses. So Staphylococcus aureus, when you have it not as on the skin, but as a systemic infection, it will start causing blood clots, and these blood clots will lodge somewhere in the circulation, and this will be the beginning of the formation of an abscess, which is specific, very specific characteristic of Staphylococcus aureus infection. DNases, often expressed by other bacteria. So why do these bacteria express DNases? Well, it comes back to this process here. Because these epithelial cells that are getting killed by the bacterial infection, they'll have a tendency to release their chromosomal DNA. So you'll have high molecular weight DNA, which is released at the site of cell death here. If it's necrotic, okay, not apoptotic cell death, necrotic cell death. And this has a tendency for the high molecular weight DNA to be very viscous, and the bacteria here can get caught up in it. So in order to escape from this initial site of infection, they need to express DNases, which once again will be an element related to dissemination from the initial site of infection. Also, yeah, since we're on this topic, okay, some pathogenic bacteria do the opposite of Staphylococcus aureus. That is, they will express fibrinolytic enzymes. So Streptococcus will express streptokinase. This one has the activity of breaking down blood clots. Okay? And once again, it's to do with the idea that once you've got an initial site of infection, these bacteria here, they will part of your response mechanism will be to form a blood clot around these bacteria. So in most cases, this will help localize the infection. So Streptococcus will express streptokinase, break down the clot, and allow the bacterium to disseminate. So Staphylococcus aureus is a bit of a weirdo here. Okay, this is the only one that will actually provoke coagulation as part of its pathogenic process. Most other bacterial pathogens do the opposite and break down blood clots. Okay, so hydrolytic enzymes, part of the colonization dissemination process, but of course they'll be directly damaging tissue structures. Now, second aspect of bacteria actively harming the host is the action of toxins. There are two types, and it's very important to know the characteristics of exotoxins and endotoxins. Very, very frequent exam question. Exotoxins are secreted proteins which will have a specific target in the host. There are different families of exotoxins, and one of the frequent ones is the AB family. So there are two components, the B, which will bind to a receptor on the host cell, and the active component, which will have enzymatic activity and will do something inside the host cell. So very, very frequently expressed, and the actions of these toxins will be specifically related to the type of symptoms that the pathogen provokes. So the big classic is cholera toxin. So Vibrio cholerae, it's a waterborne infectious agent. It's a localized infection. It's not a systemic infection, so the bacteria colonize your gut. They stay in the gut. And it's the toxin which will cause all the problems, because the AB toxin will bind to a receptor on the surface of the enterocytes. The B subunit will be internalized, and the A subunit, it's an adenylate cyclase, so it will produce cyclic AMP, which will activate the CFTR chloride channel. So this will result in an export and efflux of chloride ions into the lumen of the intestine. So to maintain osmotic balance, this will be balanced by sodium export, and then you'll lose a lot of water. And this is what directly causes the symptoms of the infection. Very profuse, watery diarrhea. And this will kill you if you don't have access to clean water to drink. So there's a very, very close link between the actions of the exotoxin, the symptoms that are provoked. And this is also useful for vaccination. And that means that infection, if you don't have the action of this toxin, will not actually be harmful. So for Clostridium tetani, the vaccine is tetanus toxoid. So it's an inactive version of the toxin. You develop antibodies against it. And the vaccination works not because it prevents the infection, but because it prevents the action of the exotoxin. Endotoxins. This is LPS, lipopolysaccharide. So exotoxins, secreted proteins, can be secreted by gram-positives or gram-negatives. LPS is only a factor in gram-negative infections. So what is happening here is when you have hyperstimulation of the innate immune response, because you've got a lot of lipopolysaccharide all over the place. So how does this occur? Well, as I said, LPS, the core oligosaccharide. Is a conserved structure in the gram-negative cell wall, and it can be recognized by receptors of the innate immune system. And when this happens, you get inflammation. Now, locally, this is very useful. What will happen during inflammation at a site where you have a gram-negative infection? You'll have lower blood pressure locally, and you'll have increased permeability of vascularization, and you'll have activation of the blood coagulation system, the coagulation cascade. Locally, this is very, very useful because it means you have lower blood flow, more vascular permeability. Neutrophils, phagocytes will be able to enter the site of the infection more effectively. You have coagulation will occur, so you'll get a blood clot. This will prevent the bacteria from disseminating. On a local level, this is all very good to have an effective antibacterial response. The problem is when you've got bacteria all over your body, all over your blood system, all through your circulation, and you have the same effects throughout your body at the same time. So you'll have very, very low blood pressure. Your blood pressure will go down like this. Blood flow will be less efficient, so you can have insufficient irrigation of vital organs, and you'll have activation of your blood coagulation all through your body. So it's called disseminated intravascular coagulation. Altogether, this will give you septic shock, and you can die because of this. So if you've got gram-negative septicemia with a lot of bacteria throughout the body, it's endotoxins in the bacterial cell wall which can directly cause this pathology and kill you. However, this is going to be not very specific, and you'll have the same kind of thing happening for all gram-negative infections. It's not the case for exotoxins, because each exotoxin will have its own specific effects. Okay. Right, so that's a summary so far. We have about half an hour left. Okay, we haven't done this bit. This comes at the end, actually. So you have to know all these points. Yeah, okay, let's take a minute before we move on to try and think about some of these things. So okay, everybody okay with this? Yeah, okay, so 1. Capsule antigens are important virulence factors because they mediate adhesion to host cells. Who says yes? Who says no? Ah, okay, you're all wrong. This is wrong, right? Capsule antigens, they're not adhesins. They're not bacterial adhesins. They help the bacterium to avoid phagocytosis. Okay, so it's an escape from the immune system. That's why they're important. Okay, I'm so glad I asked the question. All right. So B, all pathogenic bacteria have to have high affinity siderophores to get iron. Who says yes? Who says no? Some people said yes. Okay, they all need iron, but siderophores are not the only mechanism. Okay, bind iron transport proteins or lyse erythrocytes, this kind of thing. Pathogenic bacterium loses a virulence factor coding for an exotoxin. Its LD50 will increase. Who says yes? One person. Who says no? Okay. Who? All right then. So D. Both gram-positives and gram-negatives can secrete exotoxins. True. False. Hey, it's true. It's true. Okay, exotoxins can be secreted by both gram-positives and gram-negatives. Vibrio cholera, it's gram-negative. Secretes a cholera toxin and an exotoxin. Okay. Endotoxin, only gram-negatives. Exotoxin, both. Right? So let's go back to this one. Okay. LD50 is increasing. What does that mean? Becomes more pathogenic or less pathogenic? Less pathogenic, okay? If the LD50 is low, it means very few bacteria can kill you. If the LD50 is high, you need a lot of bacteria to kill somebody. So it's less pathogenic. Okay, that's why this question is in there. So if you lose a virulence factor, your bacterium is becoming less pathogenic. So your LD50 is going to increase. That's right. This is true. So the opposite is also true. The more virulence factors a bacterium has, then the LD50 is going to go down. Okay? Everybody understand what the correct answers are? Okay. So let's go on. We have about 25 minutes for a couple of specific examples. 1 gram positive, 1 gram negative pathogen. So Streptococcus pneumoniae causes bacterial pneumonia or ear infections. You can often pick it up, in the nose of people who are perfectly healthy. So it's kind of, you would call it an opportunistic pathogen. These, of course, can be quite severe, especially the bacterial pneumonia in older people. And it can also cause meningitis. Now in France, it will be causing 5,000 to 7,000 invasive infections per year and 600 cases of bacterial meningitis. So an invasive infection means growth of the bacterium in a compartment of the body which is normally sterile. So bacterial pneumonia is an invasive infection. Meningitis is an invasive infection. Just an infection in the middle ear without any complications is not an invasive infection. Because in the middle ear, it's on the outside of your body. It's not a sterile compartment. Now, who's getting infected here? So, quite a significant pathogen worldwide, causing about 1 million deaths a year. Now, people who are going to be susceptible are very young babies, okay, less than 1 year old. Young adults like you don't have anything to worry about. Whereas as you get older, it seems like your immunity wanes and people become more susceptible. So it's a significant cause of morbidity and mortality in elderly people. And once you get over 70, then the incidence of these invasive infections definitely increases. Now, how do you, what are the characteristics of this bacterium? You can isolate it on blood agar, and then you'll have gram-positive cocci, which are catalase negative. So Staphylococcus will be catalase positive. Now, Streptococcus pneumoniae will give or will provoke alpha hemolysis. So that's this here. You can see where you've got a lot of bacteria present on the plate, you have a discoloration. Okay, so this has got sheep blood erythrocytes mixed with the agar before the plates are poured. So you have some discoloration underneath the bacterial colonies, but you don't have like a massive amount of hemolysis. This is alpha hemolysis. Beta hemolysis is a huge amount of, you would have all of this region totally lysed. So what is happening here is that you've got peroxide produced during the respiration of the bacteria, which will be, or other reactive oxygen species, which are reacting with hemoglobin. There's no real lysis of the erythrocytes here. And gamma hemolysis is apparently no hemolysis. So once you have this, then you would want to identify the bacterium with an API gallery for Streptococcus. Now, colony morphology can be variable depending on the strain, and the big one that you should all know about is rough and smooth colonies. So, rough do not have a capsule antigen, and smooth, they have a capsule. And does this ring any bells for anybody? Streptococcus, pneumoniae, and rough and smooth colonies. So, ones with the capsule antigen, they are pathogenic in mice and also in humans. Colonies that have a rough morphology don't have a capsule, they are non-pathogenic. But if you mix up dead bacteria from a smooth colony and live bacteria from a rough colony and inject them into mice, it causes disease. And these live bacteria give a smooth colony morphology. This is the whose experiment? That guy, yeah. It was him. The Griffiths and Avery experiment. Okay. And part of it was to show that, and in the end they were able to show that these live bacteria were taking up DNA from the dead pathogenic bacteria, and this allowed them to become more virulent. So this is a bit unusual. Most bacteria don't do this, but Streptococcus pneumoniae is what is called naturally competent. That is, without any modification of the bacterial cell, they can take up bits of DNA and integrate them into their chromosomes. Okay? So this was an important experiment for showing that DNA is the genetic material in the 1930s, I guess. So the capsule is an important determinant of virulence. However, among Streptococcus pneumoniae that circulate in the European population, there are more than 90 different serotypes of capsule antigen, which is going to be a problem for vaccination. Except, fortunately, only a small number of these different strains are responsible for invasive infections, which are going to be causing the real clinical problems. So, since 2010, I believe, the Prevnar 13 vaccine has been available. So this has got the capsule antigens of the 13 serotypes that cause most of the invasive infections. Okay, so you don't have to have a vaccine with like 90 or 100 serotypes. You just vaccinate against the most dangerous strains. Okay, so what are the molecular mechanisms involved in virulence? So adhesion, let's start from the beginning. So you've got gram-positive cocci that are going to adhere onto the epithelium of your upper respiratory tract or in the lung to cause pneumonia. Now the receptor they use is PIGR, so the polymerized immunoglobulin receptor. So this, the PIGR, is part of your immune defense mechanisms at epithelia. So what it's going to do is pick up immunoglobulin A secreted by plasmacytes underneath the epithelium. So antibodies are going to bind on here, and then it's going to circulate up to the apical membrane, and then IgA will be released outside the epithelium. So this is good, right? This is how you get antibodies on the outside of the epithelium to prevent infection. Then what happens is this receptor will recycle and come back down to the basal membrane. So Streptococcus pneumoniae is going to use this receptor as a kind of taxi. So it will bind on the apical pole of the epithelial cell. It's going to be transported all the way down to the basal pole of the cell, and then you'll get colonization of the basal membrane here. So PSPC is a pneumococcal surface protein C. It's a non-fimbrial adhesin. And it's subverting part of the defense mechanism. Yes?
So for this receptor, it transports the antibodies from the basal pole to the apical pole, right?
Yeah.
So wouldn't taking that receptor to go to the basal pole just take you to where all the antibodies are?
Yeah, but if this person doesn't have any specific antibodies against this bacterium, they don't care. It's going to be against something else. Okay, and the antibodies that are going to be protective are going to be the anti-capsule antibodies. So once they've got down to the basal pole, then they express the hyaluronidase, which can degrade the extracellular matrix and allow the bacteria to grow underneath the epithelial cell layer. PSPA, okay, pneumococcal surface protein A, binds lactoferrin, so that's how they get iron. They express capsule antigens, so they're going to express a load of capsule antigens around here, and this will prevent phagocytes from coming in and effectively being able to internalize those cells. And they express an exotoxin, which is pneumolysin. So what's the mechanism of of pathogenesis? Well, okay, when these guys are growing in your lung tissue, they're destroying the epithelial cell layer, they're destroying the basement membrane, and they're lysing cells. So that's the pathogenic mechanism here. Second example, about 15 minutes, for plague bacterium, Yersinia pestis. So kind of like with a smallpox virus, I tried to pick one which has been very important historically. So there have been 3 big waves of plague that we know about. Okay, so the first one started out in the 6th century in Alexandria and was transferred to Constantinople and the rest of the Mediterranean region. From there it spread to the Middle East and was also ended up causing disease in China. So we know that this epidemic wave went all the way from the Mediterranean region to East Asia. There were sporadic outbreaks all the way through the 7th and to the 8th century, and then it seemed to disappear. Historically, this was very important because Justinian and his general Belisarius, they had just managed to reconquer Rome. So they were kind of working up to reunifying the Roman Empire from Constantinople. And then all of a sudden, the plague hits. 1/3 of the population is killed. And that's not really going to help any empire building process. More likely to try to kind of weaken these big big social structures. So disappears and then reappears again, in Europe at least, with the Black Death in the 14th century. And then once again, there's a huge wave of infection. 1/3 of the population of Europe probably killed during over 1 or 2 decades. And then periodically came back with plague outbreaks up to about the 18th century. And then contemporary plague, because it still exists, seems to have emerged again in the 19th century, and there's still outbreaks up to the present day. So from 2010 to 2015, about 3,000 cases worldwide and about 600 deaths. So here the case fatality ratio, CFR, is 18%. In the modern world, when people are getting treated by antibiotics. So this CFR is lower than in the historical outbreaks. Okay, so what is happening here? This is really a zoonosis. That is to say, it's an infection that has an animal reservoir that can spread to humans. So this will be rodents, either wild rodents living somewhere, in forests, in farms, but in the case of the Black Death, probably the Justinian plague as well, it would be rats living in cities. The disease, the bacterium is spread from the animal reservoir to humans by flea bites, and that is going to be important for virulence factors and for pathology. So what you have is you get bitten by a flea somewhere. Then the bacterium is going to escape from the initial site of infection and will infect the draining lymph node. It will be able to multiply, cause a lot of local inflammation, so you get a very large, enlarged, painful, inflamed lymph node called a bubo. That's maybe 1 to 2 weeks after infection. I should check my notes on this actually. Oh, 2 to 8 days. Okay. Okay, 2 to 8 days after infection. So once you've got a bubo, two things are going to happen. Either you'll be lucky, and that's maybe 40 to 70% of infection of bubonic plague cases will resolve spontaneously. You'll be able to control the infection locally and you'll recover. Okay? So Emperor Justinian, he was one of those. Okay? He got infected and recovered. Or you won't be able to control the infection in your inflamed lymph node. So you'll get, maybe 4 days later after the appearance of the bubo, you'll have a rapid deterioration of your clinical condition, vomiting, very, very high fever. And this is because you've got bacteremia. You've got bacteria all the way through your blood. And the number here is very, very high. You have more than 1 million per ml bacteria in your blood. That's important for transmission because in order for the flea to pick up the infection again, it's got to have a lot in the blood. Okay, so there's no way around for the pathogen to get transmitted unless it's multiplying a lot. But of course, this is going to kill you because you'll have septic shock. So that's going to be the pathogenic mechanism here, just very, very high bacterial load all the way through your body. So if you get to this stage, then you're going to die. Okay, so that's going to be 30 to 60% case fatality ratio of untreated bubonic plague. What can also happen is you can get pneumonic plague, so the bacterium can reach your lungs. Now there's two different meanings of pneumonic plague. One of them is when it's secondary to bacteremia. So you get infection in the blood, then you get the lungs, and in that case, 100% mortality, you die. But you can also then have transmission from direct transmission from one infected person to the other without going through with the fleas. So in that case, the initial site of infection will be the lung. That's also called pneumonic plague, but it's less severe than secondary pneumonic plague. Okay, now there's currently an outbreak of plague in Madagascar. So far there have been 849 cases. This was, last week. 67 deaths with a case fatality ratio quite low, 8%. So of course, there's an outbreak response, so anybody who's diagnosed is going to be treated with, I believe, fluoroquinolones and gentamicin as the antibiotics. And the breakdown here is you have a lot of pneumonic plague, not so much bubonic. So probably the transmission chain here is not necessarily through rodents and fleas. You have people getting a lung infection transmitting directly. Now, fortunately, I think this outbreak is come under control. So here you've got this is week by week, the number of cases per week. So this is the beginning of the outbreak, then you've got transmission, then you've got more people getting ill, and then the outbreak really starts to take hold here in September. A large number of cases, and then you've got big outbreak response from the World Health Organization, Africa region, sending in more resources, more people to try and track down the contacts of people who've been ill, treat them preventively, and this kind of stuff. So now you've got the outbreak which is under control. Okay, so characteristics of, we're going to be running late. So what are the characteristics of Yersinia pestis? Gram-negative, small bacilli, catalase plus, oxidase neg, lactase negative, so they're not colibacilli. So this was isolement in the French version. Isolation on blood agar. And or selective CIN. So CIN agar, this has got cephalosporins because Yersinia are naturally resistant to cephalosporins, and N is another antibiotic which will inhibit the growth of gram-positives. So you can grow them up on selective agar. But the problem is, this takes 48 to 72 hours. If you've got a suspicion of bubonic plague, you can't wait 3 days before you start the antibiotic treatment. The guy's going to be dead. Right? So this is not very useful for rapid diagnosis. So rapid diagnostics can be done either with, performing a gram stain on a blood sample, or there are tests for the presence of the F1 antigen, which we'll see in the next slide, because this is only present in Yersinia pestis infection, and is produced in large quantities. So if you get positive diagnosis with the rapid test, start the antibiotics. Yes? Oh, okay. Okay, bacilli look like this. Coccobacilli, kind of like oval, but it's not really spherical. Okay. Coccobacilli, coliform, this kind of thing. So not really very elongated. Okay. Yersinia pestis, what makes it so bad? Okay, so adhesion. So the main adhesion molecule for Yersinia pestis is AIL, for adhesion invasion locus. It's a non-Fimbrial adhesin. It's not really specific for Yersinia pestis because this is present in other Yersinia species, Yersinia enterocolytica, tuberculosis. But in Yersinia pestis, this is the main adhesion molecule. How do they get iron? Okay, Yersinia bactin, it produces a very high affinity iron siderophore. How do they avoid being phagocytosed? There's a very, very neat system that all the Yersinia have. It's a type 3 secretion system. So this is kind of like a complicated needle structure in the plasma membrane and the cell wall of the bacteria, which will inject these YOP proteins into phagocytes. So you've got a phagocytic cell, which is going to try and approach Yersinia and internalize it and kill it. So Yersinia, what it's going to do with the TSS, T3SS and the YOBs, it's going to inject bacterial proteins into the phagocyte, which will prevent its activation, so it's not going to provoke inflammation, and it will induce apoptosis in the phagocytic cell. So it's going to kill the phagocyte with this mechanism. However, these three things, this one, the iron siderophore and T3SS YOBs, they are not specific for Yersinia pestis. So what are specific are capsule-like antigen F1. So this is what's used for the rapid diagnostic test. So this is a Fimbrial protein, which is kind of unusual because it's going to be produced in very large quantities. It's going to cover the whole surface of the bacterium, and it's also secreted. So it has the same properties as a capsule because it will inhibit phagocytosis. Yeah. Okay. None of these things are going to be 100% effective, right? And there are different types of phagocytic cells in the body. And actually, I don't really know why, but it's very, very important for transmission from the flea bite. So in the mouse model, you've got a mouse model where you can inject Yersinia pestis under the skin and see what happens afterwards. So if you use bacteria where you've inactivated the F1 protein, then it's less pathogenic. Less important than this one, I would say, because this one, if you do the same experiment, you'll have 1 million times less. The LD50 will be about 1 million times higher if you eliminate the plasminogen activator. So this is going to degrade blood clots. Okay, so it's very important in dissemination from the initial site of the bite to the lymph node. And if they don't have this factor here, then transmission from the bite to cause a systemic infection is very, very inefficient. So these two are very specific for Yersinia pestis. Okay, just before we stop, I should say one or two things about the genetics of virulence. So the thing that is differentiating pathogens, pathogenic bacteria from commensal bacteria, is the fact that pathogens have got specific molecular mechanisms for adhesion, colonizing the host, producing toxins, producing hydrolytic enzymes. Each one of these systems is going to be several genes working together. Okay, so for an AB toxin, at least you need 2 different genes, one coding for A, one coding for B. For a siderophore, you need maybe 2 enzymes producing the siderophore and a receptor. So you have several genes and they all have to be expressed together. So virulence genes, they're non-essential. They're often organized in operons. So you have several genes all together on the same stretch of DNA that will need to be expressed together to perform the function. And they are coded most of the time on mobile DNA elements. So these are either plasmids. So for Yersinia, Yersinia pestis has got 3 plasmids. This one is shared with other Yersinia species. PMT1 and PPCB1 are specific to Yersinia pestis. So this is how this disease pathogen has evolved in time. Okay, it's come from a precursor, another Yersinia species that already had these virulence factors, and somehow it's picked up these 2 plasmids. And these give it the ability to be transmitted by the flea bite. Okay. So some virulence genes are on plasmids, others are on chromosomes. And without talking about this too much, chromosomal pathogenicity islands often are mobile DNA elements. So they can transpose, they can be lost, they can be integrated into the chromosome. They are not stable. Okay, so that's it, I guess. We're finished. Probably someone else needs to be in the room right now. So These are the things to remember about bacterial pathogens. And I'll see you all in the lab Monday after the holidays.